We all saw how the obesity rates increased since 1985 to 2 years ago. That's the most recent data. Right now, people who are obese are probably more than 30% of us. I'm not talking about overweight people, but obese. That's over 30 points of BMI, what we call body mass index. But today I will talk about another aspect of obesity, and that's how maternal obesity influences the fetal brain development. So in other words, I will end up this presentation with a question that I will ask you to think about, at least for a day. So today, we have a better understanding of how nutrition can interact with our makeup, our genes. And today, I will talk more about epigenetics and about how maternal diets can alter this brain development of the child while the child is in utero. And I will talk how these epigenetic changes will lead to gene activation, to changes in gene activation, and finally to changes in the way we function. You should also know that medical expenditures due to obesity that were equaled around 92 billions in 2002. If the trend maintains in the increase of obesity, by 2011, we will have to spend $344 billion on everything that is related to obesity. These are not exaggerations. These are families which are obese, parents and children, and of course, this child has a remote controller in the hand. Now, all of us know about the vicious circle that promotes obesity in children. A normal child, a healthy child, begins playing with video games, starts eating a little bit more fatty food, fattier food, then starts gaining in weight. Now, extra 10 pounds will make him or her moderately obese child that will let her or him to run more difficult. So the child will choose to run less. Furthermore, extra increase in body weight will lead to an adult obese, morbidly obese, meaning that will have a lot of diseases associated and initiated by obesity. But this is not the only story. This is an incomplete story. What I'm talking today about will be how this obese adult, specifically a woman, a pregnant woman, will influence how her child will develop in terms of fetal brain development. These again are charts showing adult obesity in 2009 versus 1991, when you see that most of us are around, in most states, we have 25 to 30% of people who are obese. The children lead follow this trend. We have increased in obesity in children in especially southern states. They are the highest. They have the highest rates. So what is missing in this picture? We know that whatever we do, we increase the obesity rates. Whatever we hear, whatever policies we want to follow, we still increase obesity rates. Probably something is missing here, and this is the story about a portion of what is missing, and I think we are on the right track here. I will talk a little bit about epigenetics, a little bit of history. During the human history, people realized that we interact with the environment, and Lamarck, as early as like almost 300 years ago, realized that an organism can pass on characteristics that it acquired during its lifetime to its offspring. And definitely this is not what we call genetic inheritance, because you cannot change your own DNA sequence from a generation to another. So clearly, this was something different. Later, Conrad Weddington clarified a little bit this term, and he thought about development and inheritance in terms of the crosstalk between genes and the environment, again, without changing the DNA sequence. So how is this possible? Now, we all know that we have 46 chromosomes. The DNA is packed in 46 chromosomes, 23 coming from mother, 23 coming from the father. If we go further and look closer, we will see that this DNA in the chromosome is tightly packed around what we call histones. This is a protein core made up of histones around which the DNA is packed. If we go further, closer, we will see that these histones have tails composed of amino acid sequences, and the DNA does not represent only a sequence that codes genetic information, but that the DNA can be also chemically modified by what we call DNA methylation. Again, DNA methylation means a chemical change in the DNA without changing the DNA sequence, meaning the coding sequence. But why is this important? A normal gene, like this one, the yellow-green portion, it's a DNA sequence that will code for a certain protein. Now, in order for this gene to be activated, a small DNA sequence ahead of the gene, which we call promoter, will have to be bound by some proteins called transcription factors. Now, these transcription factors will be able to activate this gene. What will happen further is that we produce what we call RNA and then a protein based on the template that was coded in this gene. If the DNA is methylated at this promoter region, then these transcription factors, which normally should bind to the promoters, are prevented for binding to this promoter. So therefore, this gene will not be active. And between these two extremes, I have a multitude of choices. So you'll have to remember that the more a promoter of a gene is methylated, the less that gene will be activated. Okay. Now, I'll talk about the focus of my research. I'm interested to see what happens in the hippocampus. That's the brain area that is associated with memory and learning. And here, this is a chart that shows the growing rates for the hippocampus using a mouse model. The mouse gestation is for 20 days, while the human gestation is for 9 months. So here you can see that the hippocampus has a higher growing rate between the gestation days 14 and 15, and then there is a second peak of growing rate just around birth and a little bit after. This is important that I will ask you to remember a bit that this hippocampus has 2 peaks of growing rates. We wanted to see whether maternal obesity alters fetal brain development, and we asked whether there are changes in the fetal brains that were exposed to maternal obesity. We wanted to start identifying pathways that are potentially relevant to what is happening and to characterize epigenetic changes, meaning DNA methylation changes. Our hypothesis was that maternal obesity will alter neurogenesis and that these alterations are associated with epigenetic changes. A little bit about study design. We took mice that were healthy, adult mice, females, and put them for a 10-week period on a high-fat diet. So they became obese. Then they were bred with males, normal males, and at the gestation day 17, we sacrificed both the mothers and the fetuses, and we took out the fetal brains and looked at that. We had also a control group where the mice were fed a controlled diet, a diet that was low in fat. What happened is that if you look here, you see the growth of body weight for the females that were exposed to a high-fat diet. So they gain in 10 weeks approximately 50% more than what the control mice gained, meaning they had probably around 31 grams on average compared to 22 grams for the control mice. So they were 50% fatter than the control mice. Now, at this point, we sacrificed, they began to get pregnant, and then after 17 days, we sacrificed the females. So what we get here is that both groups had the same number of fetuses, around 8. But in the high-fat group, the obese group, roughly 1 out of the 8 fetuses was resorbed. Resorbed meaning it was not there. Also, the body weight of the fetuses that were viable, the other 7 on average per liter, was less than the body weight in the control group. So we had smaller fetuses, and 1 out of 8 were resorbed. And this is how a resorbed fetus looks like. This is a normal fetus, a viable fetus for this gestation age. And this is a placenta. 1 out of the 8 fetuses in a mother, instead of being a fetus, we were able to find only the placenta. So we don't know whether this fetus did not develop or it developed and then it was resorbed and only the placenta remained. So we looked at the hippocampus of these fetuses, and I would like to point out that an organ grows because the cells divide in that organ, so I have an increase in the number of the cells. So this is why during gestation the organs grow. We looked at the hippocampus, and we noticed that the fetal brains that were coming from obese mothers had more cell proliferation. So more cells were dividing in the hippocampus of fetuses coming from the obese mothers. And this is in the hippocampus, not only the hippocampus, but also in the cortex, which is the brain area that is the newest one. So they had more cell proliferation. Apparently, they were developing at a higher speed. However, when we look at cell death is used after we reach the required number of cells. We start trimming the cells because we don't need all the cells. So we have to start getting rid of the cells. The apoptosis, the cell death, was also was reduced in the fetal brains coming from obese mothers. So on one hand, we have more cell proliferation. On the other hand, we have less cell death. When we look at how many cells are maturing into neurons, mature neurons, we saw that those from the high-fat group had less mature neurons. So all this indicated to us, although this is still a hypothesis, we need more research on it, that the hippocampus in the high-fat group was actually in a developmental delay, meaning that at day 17, it looked at an earlier day because it had a higher rate of proliferation, and it had conversely a lower rate of maturation. So this was our hypothesis that we have to do here with a developmental delay that is due either to maternal obesity or to a maternal high-fat diet. We have to look more into this. When we take all these brains, the whole brains, and we measure the gene expression, how many genes out of around 20,000 mouse genes are activated, we see that all the genes that have to do with these processes were down in the high-fat brains, the fetal brains. That means and suggests that the hypothesized developmental brain delay that we saw in the hippocampus may be present in the whole brain. All these processes have to do with the functioning of the brain. And the genes that are responsible for these roles were down in the high-fat brains, fetal brains. So it seems to be a general process that happens in the whole brain and not only in the hippocampus. When we looked at the genes that were changed in their DNA methylation, because we wanted to see whether there is an epigenetic connection between the maternal diet and the fetal brain development, we noticed that roughly, I think there were 18 of the genes were changed in their DNA methylation status when we compare it with the control group. And most of them had less DNA methylation than the control group, except for one. This means that we do have epigenetic changes that are triggered in the fetal brain by maternal obesity or by the high-fat diet. The conclusion for this study was that maternal obesity or a high-fat diet will alter fetal brain development, the proliferation, programmed cell death, and early neuronal maturation. That there are epigenetic changes that accompany these alterations, and that the maternal obesity negatively influences the activation of genes required for brain development throughout the whole brain, the whole fetal brain. Now, in the end, I would like to expand a little bit this discussion about what do we know when we talk about how to approach this challenge, how to reduce the obesity rates? Because obviously this bit of information today has indicated that it is bad, not only for you, but also for your child. And these alterations start in utero. We know that early obesity and comorbidities in children can be initiated during their intrauterine life by maternal diet. We also know, and it wasn't the subject of today's presentation, that third generation may be also affected. And I will get back to you a little bit later. That some alterations are epigenetically driven, DNA methylation in our instance. We have compelling evidence indicating that the obesity epidemic is caused by cumulative factors, starting with parental and early nutrition. And that once occurred, we know that epigenetic changes are long-lasting. So it is very difficult to start changing them back. That epigenetic alterations also occur in all life stages, but they are targeted and stage-specific. This means that I cannot repair the changes, the deleterious changes in DNA methylation that appeared in the fetal development. I cannot do this at a later age. What is missing in this approach to the early prevention? We do not have obesity risk, an assessment of obesity risk for subsequent generations. In other words, if I'm obese, I do not have any research right now that will show what is the risk for the third generation in becoming obese, for my grandchildren. We have to use epigenetic markers to address this risk. Some people are prone to obesity, some people are resistant to obesity, even if they eat a high-fat diet. We know, all of us, we have around us cases where we know people eating a lot of high-fat diet, but they remain lean. Do we have to care for them, or can we put them aside and focus on those that are at risk of becoming obese? We also do not have prevention policies. We know that today the prevention policy making is blind. We recommend the same thing to all people. We don't have an individualized recommendation that is based on a genomic triad, what we call it today. That is how variable our DNA code is, how many genes, identical genes we have, and what differences in the epigenetic status are between individuals. We need more human studies to expand and clarify these initial findings and to identify epigenetic markers for assessing obesity risk versus individualized medical counsel, and finding the mechanism to counteract when these changes are already there. And I will leave you with this question. for how many generations are we responsible. This slide shows you that a mother, the lifestyle of a mother may influence not only her own child, but also because in this child, in this fetus, there are cells that will become either sperm or ovules. This means that the mom can influence directly the third generation, her grandchildren, and so forth and so on. So we do not know, and I will ask you to think a bit for how many generations are you responsible when you do not eat right. Well, thank you very much, and I'm waiting for your questions. Oh, and I'd like to thank Beverly Jordan first for the wonderful work she's doing, to Daniel Lupu, my hero in the lab, who's taking care of everything. He's very talented. Steven Zeisel, and to Jenna Harrison and all the staff members that are doing our job very easy and pleasant. Thank you very much. So the question is, right. So the question is whether there is any evidence that we have that we go beyond the third generation. There is evidence in animals that we go beyond the third generation. Actually, the third generation is the last generation that can be influenced directly by the mother. From the fourth generation on, the fourth generation doesn't have any contact with that mother. But we have evidence in the mouse models. Right, when we talk about overeating, we see this risk that the fourth generation to still be to eat more when they come from an initial mother that was obese. Yes?
I've heard the speech is about epigenetics, but tonight I got a little confused. Is methylation the only way that an epigenetic change occurs?
No.
The first time tonight I heard the possibility that an external influence could cause the expression of a gene different, but may not be caused epigenetically. Did I get that right?
Well, there are many ways that the environment can influence how the genes work. And these, when I talk about epigenetics, it means that this DNA methylation, for instance, this is an epigenetic mechanism. A gene can be more methylated or less methylated than it should be according to the environmental triggers that we receive. So, for instance, if I eat more choline, this is a methyl donor that will make me more, prone to have a better methylation of the genes because I do have the stuff, I have the methyl donors there that are easier to be added to the DNA. I don't know if I answered to your question. The DNA methylation is one half of the story. Those histones, those proteins that around which DNA wraps also can be methylated or chemically changed in other ways, acetylation, ubiquitination, and other things, and those are also epigenetic processes. But the only way the information travels from one generation to another is via DNA methylation, not via histone changes. Yes, thank you.
I'm not sure exactly how to ask this. You had a list of all the things that changed in the brain.
Right.
Is that an IQ? What does it mean? With all the
These?
that? Yeah.
Well,
what is that?
I was looking at 20,000 genes, and I found thousands of genes changing, so I had to make sense of this. My brain cannot comprehend what those thousands of genes that are changed would mean for the functionality of the brain. So I had to classify them according to their roles. So, for instance, and this is called gene ontology analysis, meaning that if I have more genes that were changed that pertain to a process, I will call that process that is significantly changed, and it's of interest for me because I have to look at that process more than randomly choosing whatever process I want. So if many genes were changed for this category, neuron projection, I will go there and see, maybe look further, what it happens. But I know that all those genes that were changed in the high-fat brains pertained to these classes that have to do with how the brain works. And if you look here, we are talking about an adult brain, how an adult brain works. So all these genes that are in charge with how an adult brain works were down, meaning that I have a less developed brain. That is what this table tells me, actually.
Maybe you can explain to the layman what cellular morphogenesis, what the classifications mean in layman's terms. Neurogenesis, what does that mean?
Yeah, these processes mean, for instance, neurogenesis means the birth of neurons. How the neurons become adult neurons, because the adult neurons are coming out from progenitor cells. So in the fetal brain, I do not have only neurons, adult neurons. I have many progenitor cells. Some of them will become adult neurons, some of them will stay as a progenitor cells. So all these processes were inhibited, meaning that I have a less developed brain.
And this is when this was 17 days.
17 days during gestation still.
So does it mean you're slower or you're not going to?
They seem to be slower. They were at day 17, their hippocampus was like it would have been at day 15 or 16. Going back here, they still have more rate of growth. But this bigger rate of growth means that the hippocampus is on an earlier stage, when it grew faster.
I guess I'm trying to figure out what does it look like. I mean, you got an overweight mother.
I have an overweight.
You have an overweight kid.
I can tell you, well, the kid was smaller, would have been smaller if this trend would continue after birth. These were still in utero, so these were fetuses. But we know today in human studies that the first child born in an obese mother has a better chance, a higher chance to be smaller. And this is true only for the firstborn. The others, from the second one and on, are having higher chances to be bigger than normal.
Bigger means heavier or taller?
Heavier and longer. Bigger. Okay.
Question. You might think I'm stupid when I ask it, but when they do a lot of these studies, they use mice. Why do they use mice? Are that mice more like people or what?
It's a very good question.
I mean, I've always wondered.
We use mice because it's convenient. It's cheaper. We can study them in months. That is the equivalent of years in humans. The best way probably would be to study monkeys, Macacus rhesus, for instance, and we have such studies. But these are very expensive studies and take a longer time, much longer. So this is why we choose rodents, mice or rats. The other half of the coin, the other face of the coin, is that mice do not behave necessarily like humans. So not everything that we find in mice will apply to humans. And this is where we need to confirm later in humans whether this is true or not. Yes?
In your mouse study, with the time period that you were noticing the change in the brain at 15, 17 days,
Mm-hmm.
approximately, what would that equate to for a human being?
To the third trimester.
Third trimester.
Because the gestation is for 20 days in mice. So That would be the third trimester, the fourth quarter, sort of. Towards the end, when the brain starts to develop and starts to shape its own brain-specific areas.
How soon do you, well, I'm just curious, how would you identify an obese-resistant individual versus, I guess, a non-obese resistant individual, those who are non-sensitive? I mean, would it be a blood test? Would it be a mouth swab?
Well, the question is a very good question. How would I identify at the beginning, before becoming obese, an individual who is either resistant to becoming obese or prone to obesity? We do not have such markers. I'm working at a project, for instance, where, because not all these mice became obese when I put them on a high-fat diet. Only 60% became obese. The other 40% remained within the body weight of the control group. So they were definitely, I had 40% of mice who were resistant to obesity. And this happens to rats also. So what I want to see is whether some epigenetic changes at the beginning can differentiate them and predict whether they will become obese or not when put on a high-fat diet. That's another project I'm working at.
Did they eat less?
I don't know yet. 60% calories coming from fat, and the fat was mainly lard, so a very bad one. Yeah. Yes.
What would you say is the take-home message for the obstetrician's office when counseling his clientele?
I think regardless, with regard to this presentation or independent of this presentation, I would say eat less calories, but being careful to have all the nutrients that are recommended.
So would you recommend a diet of 30% fat or less?
30% fat is a very high fat diet for humans,
Okay.
in calories, when we talk about calories.
Then what percent?
Well, it depends. I think this is where it comes to the individualized nutrition. I would say that we have to go towards this thinking that we can't just simply recommend the same thing to everyone.
How soon do you think we'll be ready for the genomic triad?
Well, we do research. We have studies that in part are looking for each of these components. We have studies which are looking at genomic variability, at copy number variations, and epigenetics. But by now, how soon? I think it depends of how money we'll have for research, actually, because these are complex studies.
And expensive, I would imagine.
Very expensive.
The 40% of mice that had the high-fat diet that did not become obese, were they included in the study, or was it just the
No, they were not included in the study right now. Yes, so the question was whether the mice which were resistant to obesity were included in this study. They were not, because I wanted to have pregnant dams that were obese.
So we still don't know yet if it's the obesity or just the high-fat?
I don't know. I need a timed study. Right. Yes.
Your slide indicates that there is a delay in neural formation.
Mm-hmm.
In many cases, is there a finish line in time? In other words, they're delayed and they can never catch up, or is it possible that they're delayed but their maturation continues longer and they arrive at the same end state?
So the question is whether this delay, they will catch up or not. I know about a study in mice, the very same mouse strain, where they see these changes. They didn't look at the fetal changes, but they look after birth. And they saw these changes until the postnatal day 70. And the mice are living for, it depends, 1.5 years or 2 years. So they went so far to look until postnatal day 70.
And there was no improvement in their condition.
They, at day 70, they became equal in terms of how their hippocampus was developed with the control group. But they do not have any behavioral testing to see whether there are still behavioral changes or not beyond that period. 70 after birth, yeah.
Do mice show leptin resistance and sensitivity like humans? That would equate to 40%?
I did not look at leptin resistance in this study, but for sure mice which are becoming obese have the whole metabolic syndrome and also leptin resistance, yes. And there is a strain that is leptin resistant to the OB mouse, which is constitutionally fat.
If there are no other questions, could you give us a 5-minute commercial and tell us about your book?
Oh, this is I'm editing a book that is entitled, I think, Nutrition and Epigenetics. I'm only the editor of this book, but I would like to present it as an international effort. We have authors writing chapters, authors which are coming from Australia, France, Germany, UK, Canada, and other countries. This is I think it's the second book that deals with nutrition and epigenetics, and I encourage people to read it. It's not such a easy to read book, but I think it could be fun.
When's it coming out?
Late spring.
So next year you can have a speech and a book signing.
Well, I hope to have it this spring.
This spring.
Yes, this spring is yeah.